physical versus linkage maps. As I told you that we must be aware that what is the difference between these two maps. जैसे कि in case of linkage map, the relative position of the gene can be known and relative distances. Whereas on the other hand, physical maps not only pinpoint the certain features or location of the genes, but it also gives distances from one genetic marker to another in terms of the number of nucleotide bases that are present between them. So now here we can compare these two maps. Initially, around the early 20th century, most of the maps that were developed were genetic linkage maps on the basis of pattern of inheritance of certain traits. Then with the passage of time, when new techniques were available for sequencing, digestion of the genomic DNA, electrophoresis, then scientists constructed more detailed map, that is physical maps. Now if we take the example of very simple microbes like E. coli or bacteria, number of maps have been obtained and different techniques have been used, like one can use the interrupted mapping, recombination mapping, the process of transformation, where microbes, they can take naked DNA from the environment, a transduction process where some vectors like the bacteriophages, they can transfer DNA from one host to an other host or other microbe. But most of the maps that were developed for bacteria, they were based on interrupted mating. So what happens during the process of conjugation, if you remember that the DNA from the donor, it is shifted to the recipient by the formation of a conjugation tube. Now, scientists have developed certain strategies by which they can interrupt this process of mating after different time intervals. So, interrupted mating is a process by which the sequence of transfer of genes from donor to recipient, it can be studied after interrupting the mating process at different time intervals. तो हम ये स्टडी कर सकते हैं कि अच्छा इतने मिनट बाद ये जीन ट्रांसफ़र हुई है या इतने मिनट के बाद ये करैक्टर अब ट्रांसफ़र हो गया है, और आफ्टर टाइम इंटरवल्स हम जो मेटिंग है या कॉन्जुगेशन का जो प्रोसेस है बिटवीन द डोनर एंड रेसिपिएंट उसको इंटरप्ट करते हैं, एंड दिस प्रोसेस इट इज़ कॉल्ड एज द इंटरप्टेड मेटिंग, and it is widely used to map the bacterial chromosome or to generate the physical maps. अगर हम E. coli की एग्जांपल लें तो जो पहला जेनेटिक लिंकेज मैप कंस्ट्रक्ट किया गया था दैट वाज अराउंड 1963, और उस मैप के अंदर अराउंड द पोजीशन ऑफ 100 genes was given. Then after 27 years, around 1990, by using more advanced technique, around the position of 1400 genes, it is known today, because now it is more elaborated. And then with the passage of time, we have also developed certain techniques where we can compare these two types of maps, that is linkage maps with physical maps. As I told you that if the linkage maps they have constructed earlier, now we can know the accuracy. कि अगर हमने कोई 25, 30, 40 साल पहले कोई जो मैप्स बनाए थे जिनको हम लिंकेज मैप कहते हैं, जहां हमने ये अज्यूम किया था कि डीएनए के ऊपर जो जीन्स हैं इस जगह लोकेट करेंगे या पर्टिकुलर ट्रेट की पोजीशन ये होगी, अगर हम उसको आजकल की मॉडर्न टेक्निक्स को यूज़ करते हुए उनको हम फिजिकल मैप से कंपेयर करें तो क्या सिचुएशन आएगी. तो ये देखा गया है कि इन मोस्ट ऑफ द केसेस द कंपैरिजन इट इज शोइंग द मैचिंग, दैट the relative position of the genes that was assigned on the DNA, it is the same as in case of the physical maps. For example, it can be illustrated with the help of a few diagrams. So this is the map of E. coli that was constructed in 1963, and it is showing the location of around 100 genes on the chromosome. And this map was constructed by using a technique which is called as the interrupted mating. And here to the internal circle, we have time where the interruption was made, and then certain region they have further magnified here that indicates the position of different genes along the chromosome. So this was the first map that was constructed in 1963. In the next diagram, that is more or less a recent one constructed in 1990. It is showing some portion of the map that is around 5 minutes interval. So some part of the 100 minutes map is showing that is only 5 minutes. So it is also E. coli linkage map. So these different arrows here indicate the site of or the direction of transcription, and the position of different genes, it is shown along the chromosome. So now this map can give the position of around 1400 genes on the chromosome. Similarly, this diagram, it showed the comparison between the linkage map and physical map. Upper one, it is the linkage map, and lower one, it is the physical map, where we have the exact idea that where a particular gene or genetic marker it is located. So if we compare these two regions, we will know that most of the part they are showing matching after several years. So if we consider the lower diagrams, the certain red areas, they indicate the position of some regulatory genes, and some blue genes, they indicate the genes that are involved in DNA replication, recombination, etc. So here we may have the idea that certain genes like this one in the linkage map, it is the same as on the physical map, and like here this gene and this gene, it is the same. So we can say that linkage maps that were developed earlier, they are showing similarity with the physical maps.